Hello, my name is Nicole Mullen, and I am an undergraduate researcher in the Doe Lab at the University of Oregon. My research mentor is Kristen Lee, and we are investigating the role of homeodomain transcription factors in neuronal circuit function in Drosophila. Throughout my poster, I will refer to homeodomain transcription factors as HDTFs. HDTFs are proteins that control gene expression and are important for neuronal development. Previous research has shown that HDTFs are uniquely expressed in neurons to generate neuron types during development, but their role in establishing neuronal circuits remains unknown. A neuronal circuit is formed when two or more neurons begin communicating with each other. In this study, we chose to focus on the well-characterized MDN locomotor circuit in Drosophila, seen in Figure 1. The Moonwalker descending neuron, MDN, seen in Figure 2, is required for backward locomotion. Thus, when MDN is activated, the flies move backward. To study HDTFs, we activated MDN, tracked the fly's behavior, and determined if knocking down a specific HDTF causes a change in backward locomotion. Changes in behavior could suggest that HDTFs are important for this circuit. We used the GAL4 UAS system and optogenetics to accomplish this. The GAL4 protein is regulated by a neuron-specific enhancer. When the GAL4 protein is present, it leads to the transcription of the transgenes following the upstream activation sequence. As seen in Figure 3, we are expressing the GAL4 specifically in MDN. We are using UAS transgenes to knock down the HDTFs and activate MDN. To knock down a specific HDTF, we utilize the RNAi transgenes specific to that HDTF. The control larvae did not have an RNAi transgene and therefore had normal HDTF expression. To activate MDN, we express the optogenetic cation channel, Chrimson. When fed the ATR drug, the Chrimson channel is primed, and this allows MDN to be activated during red light exposure. Larvae fed vehicle, i.e. no ATR drug, have an inactive Chrimson channel, so MDN is not activated upon red light stimuli. Locomotion during red light activation is tracked using the FIMTrack software. Figure 4 validates this optogenetic method. The distance traveled forward and backward during the red light is reported. The black bars represent flies fed the ATR drug, and thus MDN is activated during red light. The gray bars represent flies fed the vehicle control, and thus MDN is not activated during red light. During red light exposure, backward locomotion was enhanced and forward locomotion was blunted in the ATR-fed group compared to the vehicle-treated group. This data confirmed that optogenetically activating MDN causes a significant behavioral change in the larvae. With this data in hand, we began knocking down individual HDTFs in MDN and investigating how losing that HDTF influences the backward locomotion when MDN is activated. Analysis of the data reveals 7 HDTF knockdowns out of the 105 tested that produced a change in MDN-induced backward locomotion compared to the baseline. Figure 5 shows 3 HDTF knockdowns, represented by the purple bars, that blunt backward locomotion compared to the control larvae shown by the black bar. These HDTFs are PDM3 in panel A, LIM1 in panel B, and DFD in panel C. On the other hand, Figure 6 shows that knocking down 4 HDTFs in MDN enhanced backward locomotion. HDTF knockdowns are represented by the blue bar, and control larvae are represented by the black bars. These HDTFs are CHN in panel A, CG32532 in panel B, VSX1 in panel C, and so on in panel D. Taken together, these data suggest that these 7 HDTFs are required for normal backward locomotion and normal MDN circuit function in Drosophila. In future research, I hope to investigate potential changes in MDN morphology, molecular markers, in neurotransmitters, the number of synaptic sites, and other abnormalities when candidate HDTFs are knocked down. Then I will correlate these knockdowns with any changes of the MDN circuit development and maintenance. Thank you.